Well, good morning and good afternoon, depending on where you are, and welcome to the March 2026 edition of UW-Madison Global Health Institute monthly webinar titled Clean Water's Role in Global Health Outcomes. My name is Kaylin Ostrowski, and I have the good fortune of serving as the Associate Director of Strategic Partnerships and Development here at GHI. Thank you for joining today's discussion. For those of you who are not familiar with GHI, we are a transdisciplinary initiative dedicated to advancing health and well-being for people and the planet through research, education, and collaboration. We coordinate, connect, and incubate efforts across disciplines and sectors to address the world's most pressing health challenges. Our monthly webinar series features researchers and practitioners from UW-Madison and around the world, highlighting the complexity of global health challenges and the many kinds of expertise needed to address them. Before we jump in, just a couple of quick housekeeping items. One, we are recording today's session and the archived video will be available on our website at ghi.wisc.edu. And second, we are monitoring the Q&A section, so please post any questions or comments for the panelists there. We will do our best to address as many as possible during the Q&A session. In celebration of this week's World Water Week, we'll explore the One Health connections with water, sanitation, and hygiene interventions, share current critical research and findings, and offer ideas for inspired collaboration. It is my privilege to introduce our panelists, each offering a unique perspective on the critical role of research and development in addressing clean water. We begin by welcoming our moderator, Dr. Trina McMahon. Dr. McMahon is a professor of bacteriology, a professor of civil and environmental engineering, and runs the Applied and Aquatic Microbiology McMahon Lab at UW-Madison. Her research focuses on how microbes have extraordinarily diverse and sophisticated physiologies, communication strategies, and mechanisms of evolution. The broad objective of her research is to improve our capacity to predict and model microbial behavior while searching for novel biology-mediated transformations that can be harnessed for engineering applications. She and her students study the microbial ecology of both natural and engineered systems. They use molecular tools to investigate microbial community structure and function in lakes and activated sludge. And their work on freshwater lakes leverages the North Temperate Lakes Long-Term Ecological Research Program, providing rich contextual data for the 20-plus year time series of microbial data. Welcome, and thank you for joining us today, Dr. McMahon. Next, we welcome Dr. Heather Murphy. Dr. Murphy is the Tier 2 Canada Research Chair in One Health and Associate Professor in the Department of Pathobiology at the Ontario Veterinary College within the University of Guelph. She has over 20 years of experience in water-related research in both North America and abroad. Her PhD in environmental engineering work was focused on appropriate household water treatment technologies in rural Cambodia. Following that, she worked for the United Nations International Emergency Children's Fund as a water sanitation and hygiene specialist in Mali and Madagascar. She returned to academia and completed a research fellowship with the Public Health Agency of Canada, where she focused on quantifying the burden of waterborne disease on the Canadian population. She was recently inducted into the Royal Society of Canada and their College of New Scholars and was named one of Canada's top 100 most powerful women in 2025 for her work and contributions to mentorship of female scientists. Dr. Murphy's research interests involve understanding and addressing water and health challenges in both developed and developing countries using a One Health approach and seeks to identify and understand the ways enteric pathogens move through water and the environment and propose appropriate interventions and water and sanitation control measures needed to protect human and animal health. Welcome and thank you for joining us, Dr. Murphy. Next, we welcome Nicole Weber. Nicole Weber is an affiliate faculty member at the Dornsife School of Public Health at Drexel University and co-author of the Water and Sanitation chapter in Health and Humanitarian Emergencies, Principle and Practice for Public Health and Healthcare Practitioners. She's also pursuing a PhD at the Nelson Institute for Environmental Studies at the University of Wisconsin-Madison. Nicole's professional experience includes living and working on public health and humanitarian issues in the Democratic Republic of the Congo with Mercy Corps and the Premier Orga International, as well as working in rural Chad, supporting the frontline guinea worm eradication efforts with the Carter Center. She previously served as a research fellow at the U.S. Centers for disease control, where she provided technical assistance on water and sanitation to support quality of care in healthcare facilities and deployed for emergency responses to cholera and forced mass displacement. More recently, she was a technical WASH director at Save the Children for a U.S. government-funded global WASH capacity strengthening and applied research project in support of food security and nutrition in fragile humanitarian settings. Welcome, and thank you for joining us, Nicole. And lastly, we welcome Dr. Matthew Freeman. Dr. Freeman is the Isa Griggs Candler Professor of Environmental Health with joint appointments in the Departments of Global Health and Epidemiology at Emory University. His work is on the impact of environmental drivers of infectious disease, specifically access to water, sanitation, and hygiene in low-income settings. His interests include the role of foodborne transmission through formal food value chains and other water, sanitation, and hygiene pathways, and on implementation research on designing and testing environmental health intervention strategies. He has led field trials in Mozambique, Cambodia, Mali, Kenya, India, and Laos, and elsewhere, assessing the impact of school-based, community-based, and multi-sectoral WASH improvements on diarrheal disease, respiratory infection, neglected tropical diseases, and growth shortfalls. His work in Mozambique assesses the role of chicken-related pathogens in child health, and a separate project assessing the impact of an urban water supply improvement on child microbiome and gut health. An ongoing trial in Cambodia is exploring the costs and benefits of intervention strategies to move to cleaner cooking technologies. Welcome, and thank you for joining us, Dr. Freeman. And with that, I am pleased to now hand it over to Dr. McMahon to begin today's engaging discussion. Thank you all for being here.
Thanks so much, Kaylin. It's great to be here. Everybody good? Able to see my screen? Yep. All right. Awesome. Yes, I'm Trina McMahon, and I have the pleasure of being the MC today for our panelists and the Q&A. So I'll tell you a little bit about myself and how my research relates to the topic. As Kaylin mentioned, I am a professor at the University of Wisconsin-Madison, and I study the water cycle from the perspective of microbes. I'm actually trained as an engineer. I did my degree in civil engineering at the University of Illinois, Urbana-Champaign, and stayed there for my master's, also in civil engineering, but then moved to UC Berkeley for my PhD, also in civil and environmental engineering, and came running back to the Midwest, since I am a Midwest girl at heart, and joined the faculty at UW-Madison, where I have been ever since. And around 8 years after I arrived, I split my position, so I'm 50% in civil engineering and 50% in bacteriology, which most people think is a pretty strange combination. But because I study, the interface of that engineered water environment and natural systems and how microbes, move through that system, it makes perfect sense to me. And I also interact a lot with the folks in the Center for Limnology here, and through that North Temperate Lakes LTER that Kaylin mentioned. So if you haven't been to Madison or you aren't in Madison, maybe you haven't heard of Lake Mendota, but this is what we like to call the best studied lake in the world. So limnology, which is the study of lakes, is thought to have been invented in Madison. That's a little bit of a grand claim, but certainly it's considered the birthplace of North American limnology. And this lake is embedded in our sense of place here in Madison. It's the biggest lake of a chain of lakes that the city sort of straddles. And what you're seeing here is a picture of what's called the Terrace, and it's right on the UW-Madison campus. And so you can see there's a lot of recreation and just going out and sitting on the lake and enjoying beer and bratwurst that happens here on campus is very iconic. So this is a picture of Lake Mendota from the air, and you can see here the university is kind of on this side of the shoreline of Lake Mendota. I'm hoping everybody can everybody see my thing? Okay. And downtown Madison is here on what's called the Isthmus. And what you can't see terribly well is that on the north side of the lake is a lot of agriculture. And so we have this mixed use landscape of urban, which is, a growing city of Madison, and then this agricultural use that's to the north. And there's a river that comes into the lake from the north. And if you know anything about Wisconsin, you know that we're famous for our cheese production or dairy production in general. And so a lot of those dairy farms to the north of Lake Mendota and to the north of Madison use a lot of manure as a way to both manage manure for fertilizer as a way to manage the production of manure from the dairy farms, but then also growing the crops to feed the cows. So it's an interesting loop there in terms of nutrients and agriculture. And we know that it's the manure that comes from those cows that when it's spread on the farm fields, can run off into the receiving waters from those fields and ultimately into the river that comes into Lake Mendota. So we have manure plus rain equals a lot of green. So maybe you've heard of blue-green algae before. This is a picture of a blue-green algae bloom on Lake Mendota that is senescing. So it's starting to decompose. And so you can see that bluish green color, which is where the blue-green algae name comes from, but they're actually cyanobacteria. So they're not algae, they're true bacteria. And that maybe is just a distinction that microbiologists like to make, but it does have implications for how we understand their biology. And in the summertime in Madison, we can get some pretty disgusting, what we would call harmful algal blooms formed by these cyanobacteria. And so this is a little bowl showing how thick and gross those cyanobacteria can become. Now, the problem is that, beyond just the aesthetics of seeing the blooms, these organisms are known to make toxins. And these toxins can be hepatotoxins, so affecting the liver, neurotoxins, which affect our nervous system, our brain. And so there is concern about exposure to these blooms, both for humans and for animals, both the animals that live in the lake, but then also animals like dogs that would play in the water. And every summer we have reports of dogs that get ill or even die from consuming the water when they jump into it. It's very rare to hear of humans becoming ill from recreating in the water, but that's partly because we don't have a lot of ability to make a connection between symptoms of exposure to a toxic bloom and the symptoms, and how they would manifest a few days later. Doctors aren't particularly informed about what those symptoms would be, and making that connection can be really challenging. So we actually don't have a good handle on how much this is impacting human health in the Madison area, and this is true for lakes around the world. We know that the toxins are very potent, but we don't have a good handle on how that manifests as a burden on humans that interact with those blooms. Now, in the research world and kind of the water quality management world, we ask ourselves a lot of times, how can we tell when and if these blooms are toxic? Because there is some complex biology that is embedded in the way that these organisms will respond to their environment and make toxins. And so there's a lot of active research on trying to figure out how we can forecast toxic blooms and maybe even predict how much of a toxin would be present. Now, the problem that we have is that not all cyanobacteria can make toxins. And we want to avoid telling the public to not go near the water anytime it's green, right? Because then people get this perception that we can't use our water resources, our lakes are hopelessly polluted, we can't, there's no point in trying to, manage them anymore because it's green and disgusting, right? And so we would like to be able to forecast when a bloom is making a toxin so that we can be more precise and rational in our advisories for people interacting with these waters. So in a bloom, you can have some fraction of cells that are not even capable of making toxin. So from a genetic sense, we can see whether or not they have the genes, so the genetic potential for making the toxin. But even those cells may or may not be ready to make a toxin at a given moment because those organisms, the individual cells, have to undergo a process where they make the machinery to build the toxin, and not all of them will, in a given hour or day or a given bloom event. And so you can have a fraction of those cells that are prepared to make the toxin, but then some fraction of those may only or may not actually make the toxin. And so deciding if you have a certain amount of cyanobacteria in water, how many of those are actually going to be toxic is a very challenging research question, and something that hasn't been solved, and is one of the things that my group works on. Now, another factor to consider is that the toxin stays inside of the cells as long as the cyanobacteria are alive. Once they start to die and decompose, that toxin gets released into the water and is now, exposed to environmental conditions that would actually make them break down rather quickly. So their time that it's in the water and not in the cells and still toxic is quite short, like within hours to maybe a day or two. And so understanding as the blooms senesce and decompose and when the toxins will be gone is another important research question. Now, obviously, probably the best way to determine if the water is toxic when you see this blue-green color is to go and measure the toxins. But the problem that we have is that the toxin analysis is extremely expensive. It can be over $100 per water sample, and the amount of time that it takes to make that measurement is too long to be able to make adaptive recommendations for people to use the water or to not use the water. And um, for research purposes, we can, if we have research grants, we can pay to make those measurements. And I wanted to just give you a sense of what that looks like before I wrap up. So these are the molecules that we most often encounter in cyanobacterial blooms. You can see they're complex structures. They have, weird names, which, we're not going to get into here, but they're complicated to measure because of their diverse structures. And so the tools that we use to measure them are complex and highly technical. So it takes a lot of skill to be able to do those measurements. And so this is just one piece of data that I'm going to show, and this is from Lake Mendota over 5 years, 6 years. And each row of these graphs is a different year. So we have 2013 to 2018, and in each plot, we have the toxin concentration. This is kind of like an aggregate level of micrograms per liter. So what you mainly need to know is that the World Health Organization has set a limit of 1 microgram per liter for the standard for drinking water to be safe from the perspective of cyanotoxins, and 10 microgram per liter for the perspective of recreation. So coming into contact with that water through swimming or water skiing or whatever. And we have the different kinds of toxins broken out by color. And so the total height of this chart here, where the red is peaking, would be the total concentration of toxins and its maximum point. And so you can first see that it doesn't go above 7.5 across these 6 years. So we're below the level of the recreational advisory threshold. But we are above the drinking threshold. We don't use Lake Mendota in particular for drinking water in Madison, but there are lakes like Lake Erie, which has similar kinds of blooms that do use the water for drinking. And of course, elsewhere in the world that have these problems would have this as a potential concern for drinking water. And of course, I could answer questions later about drinking water treatment and whether we have to worry about the toxins coming through in our drinking water. And what I'm also showing here, though, is what we're calling different regimes of the cyanobacterial blooms. And so in Lake Mendota and in many lakes that we have what we call succession of the organisms that make the water green, where you can have true algae, then you have other kinds of cyanobacteria, and then you have a diverse mixture of cyanobacteria. And then in when the weather gets cold, the cyanobacteria go away. And so the purpose of showing this cyanobacterial regime along the bottom of each chart is to show you that each year it's a little bit different, right? There's a sort of predictable stages of the cyanobacterial community types, but one thing we're trying to figure out is why it's different each year and how that relates to the amount of toxin present. Because you can see that there are some years that are more toxic than others, and we don't understand why that is. We know that, weather, the amount of rain that falls in the springtime and washes the manure into the rivers is one factor, but this is an active area of research that this is actually unpublished data, but we're trying to work it up into a publication to talk about what the drivers are of these differences in toxin concentration. So before I turn it over to Heather, I just wanted to give you kind of a overview of what we have learned, and that is that the more manure that we have, the more cyanobacteria that we have. And the culprit in that manure is phosphorus. And I didn't mention that earlier, but it's a specific element that ends up limiting the growth of these organisms. So any source of phosphorus going into a freshwater lake will encourage more cyanobacteria to grow. Some of those organisms that will grow can make toxins, but not all of them. And not all of them will be making that toxin on a given day. So we would love to be able to measure those toxins in the moment to be able to give the public more advice on when to avoid the water. But the best that we can do right now with the expense of that is to look in samples at a research level and try to make models and an understanding that we can use to forecast those blooms. If you look at Lake Mendota specifically as kind of a case study, we see that those toxins are pretty much always present from July to October. And we don't know what's causing that year-to-year variation, but we know that the levels are below the World Health Organization recommendations for drinking or for recreation, though not for drinking. And so I think that I have come up on my time. So I want to thank everybody for listening to me talk about my favorite topic, which is microbes. And now I'm going to have it turn it over to Heather, who will talk about another aspect of sort of world water topics. And Heather, I think you should be able to take it away.
Great. Thanks, Trina. Can everyone see my screen? Are we good that way? Thumbs up. Yeah, thanks, Matt. Thanks everyone for having me today. I'm going to talk briefly about kind of what I do in terms of using a One Health approach for waterborne pathogen reduction. And I often start out presentations with a bit of my background because I'm an engineer by training, but now I sit in a veterinary college. So I think sometimes it's helpful for people to see how I got there. So I started off doing my master's and undergraduate degrees in civil environmental engineering in Canada at Dalhousie University, which is on the east coast of the country. And through that time, I got really interested in water treatment for low-income countries. So I ended up pursuing a PhD at the University of Guelph in civil environmental engineering. And that's a picture of me doing work in rural Cambodia, where I looked at household water treatment technologies. And so when I finished my PhD, I really had no interest in staying in academia. And so I ended up working overseas at the UNICEF in Mali and Madagascar. And while I was working with UNICEF, I actually met Matt Freeman, one of our speakers today, and he was evaluating the impact of one of our studies in Mali. So he was looking at the health impact of one of our interventions. And that's when I got very interested in kind of the public health implications of the work I was doing. And so I returned to Canada and pursued a research fellowship with the Public Health Agency of Canada, where I looked at quantifying the burden of disease. And there I got experience in epidemiology and public health. And as I decided what I wanted to do with my career, I thought, well, maybe actually I could go back to academia and kind of sit at this interface of practice and research. And so I ended up at a School of Public Health at Temple University in Philadelphia, where I spent 5 years doing research and established the Water Health and Applied Microbiology Lab. And as I was in the US, I actually started wanting to go home to Canada and I started to look for positions. And funny enough, the University of Guelph and their veterinary college had a position open in One Health. And I actually had no idea what One Health meant at the time, but I read the job description and it looked at looking at interdisciplinary methods to solve reducing diseases in humans and animals. And it sounded like something that I might qualify for. And lucky for me, they picked an engineer to be in a vet school. So now I'm surrounded by veterinarians and learning new things every day. And so I'm not sure if everyone's familiar with One Health, but it's kind of this intersection of animal, human, and environmental health. And the way I describe my research is that I look at pathogens that are shed by animals that move through the environment and end up in our water supplies, as well as pathogens shed by humans that move through the environment and end up in our water. And then as a result, animals and humans are subsequently re-exposed through water through different pathways. And I work with a whole range of experts. So I work with veterinarians, biologists, microbiologists, policymakers, medical doctors, social scientists, animal producers, and public health experts. And I have a couple key areas of my research program. So one is I've continued this line of research on burden of waterborne disease, where I look at recreational waterborne disease in drinking water. But more recently at the University of Guelph, I've been looking at reduction of zoonotic diseases using a One Health approach. And so I have a project that's ongoing in Madagascar that I'll speak about briefly today, looking at lemurs, one on rotational grazing of beef cattle in Ontario, and then one on zoonotic disease reduction in Burkina Faso and Senegal, which I'll touch on very briefly today as well. So the first project that we've got ongoing is looking at mapping zoonotic disease transmission between lemurs, humans, domestic animals in rural Madagascar. And this work is being led by my PhD student, Amilcar Bizikari. So he's in the middle here. And it's in collaboration with Dr. Travis Steffens, who's a lemur expert at the University of Guelph. And these are our partners on the ground that work for Planet Madagascar. And so for those who aren't familiar, lemurs are only found in Madagascar, and 98% of them are threatened with extinction, and they're the most endangered group of animals in the world. And 31% of lemur species are actually critically endangered. And so Travis approached me when I first started at the University of Guelph and asked me if I knew anything about actually cryptosporidium in water, because he had suspected that lemurs might be dying from cryptosporidium in his field sites. And I said, well, actually, I do know about cryptosporidium. And I said, but what about other zoonotic diseases? And so we started working together and got some funding for this project to look at, well, what diseases might be circulating between lemurs, humans, and livestock. So his field site is in Encara National Park in the northwestern part of Madagascar. And in this national park, there's a lot of pressures on the environment where we have a lot of forest fires and fires purposely for having grasslands produced for agriculture. And so we see animals and humans, like their domestic animals, getting in closer proximity to lemurs and threatening their habitat. And so what we wanted to do with this research was first, we have four different objectives, but first we want to identify the most prevalent zoonotic pathogens between lemurs, domestic animals, humans, and water. The second was to try and determine what risk factors are causing zoonotic pathogen occurrence. The third is to try to map zoonotic pathogen transmission patterns in the national park and the communities that live there. And then lastly, assess local household knowledge, attitudes, and practices related to zoonotic disease transmission so we can ultimately develop interventions to reduce disease transmission. And so I'm just going to show you some very, very preliminary results. So we ran fecal samples and water samples using PCR methods as well as microscopy in the field. And to date, we found that pathogenic E. coli and cryptosporidium were found in the river samples, domestic animals, lemurs, and humans. And then via microscopy, we found that ascarids, isopora, and strongylides, different parasites, were shared between humans, lemurs, and domestic animals. And so this work is ongoing. I'm not showing all the results of there were different pathogens that were also shared between, say, humans and lemurs and domestic animals and lemurs, but these are the ones that were the most common across the different matrices. And so this work is continuing over the next couple of years. The last project I wanted to briefly touch on is a project in collaboration with the University of Montreal in Burkina Faso and Senegal. It's entitled Eluso, or Elle lutte contre les zoonoses. It's a One Health project looking at improving the lives of women and girls who raise small livestock. And we have 10 pilot projects in communities in both Burkina Faso and Senegal. And we started this project in late 2023, doing baseline data collection to understand the local context and potential zoonotic disease risks. In 2024, we started community engagement activities and developed pilot interventions with the communities, which is ongoing, and those interventions are being implemented. And then we've been doing One Health education and monitoring and evaluation, and the projects will continue through 2028. One thing I wanted to highlight and talk about today is during this project, in our baseline data collection, we used a method called participatory mapping that we adapted to kind of answer One Health questions. And it was really informative for engaging the community in what could be affecting animal and human health. So we had the communities map out their community and where everything is located. And then we had them identify separately what they think are the top 3 environmental risks to humans, and then the top 3 environmental risks to animals. And what was interesting, the communities identified on their own without any external assistance that some of the risks overlapped for animals and humans. And so they identified standing water, unprotected wells, solid waste, and animal carcass disposal, for example, as risks to human and animal health. And here's some pictures from the communities. You can see the kind of open wells, unprotected water supplies, and solid waste issues. And so what was also really interesting for us, our funder really wanted us to work on zoonotic disease reduction, but the community priorities did really not always align directly with zoonotic disease reduction. Instead, solid waste management and collection was one of the key priorities. And so in the end, that's what we focused on in some of these communities, because we don't think we're going to make it anywhere in terms of interventions looking at zoonotic disease reduction if we don't have community buy-in. And that participatory mapping process was super informative, because if we just relied on our other baseline data collection, this would have been a blind spot and we wouldn't have picked this up. Other things that the communities identified, they wanted fencing around solid waste sites to prevent animals from going in and eating the solid waste. They wanted construction of latrines in some communities that didn't have them. Improved and upgraded water supplies was a high priority. Fencing of water supplies to keep animals out. Having separation of human and animal excreta from water supplies. And then also having separate water supplies for animals and humans that were dedicated for each group of people. And so I think I'll finish with that and pass it back to Trina so she can pass it to our next speaker. Thank you.
Thanks so much, Heather. Super interesting. Nicole, do you want to take it over next?
Great. Thanks so much. Can everyone see my slides? Great. So good morning. I'm really happy to be here today. Thank you so much for the invitation. Today I'll be talking about WASH, which stands for Water, Sanitation, Hygiene, in recognition of the recent World's Water Day. I will focus on what I consider to be the 4 key messages that I think are important when we think about WASH and health outcomes. So briefly, as the bio was already read, I would just say that most of my career has focused on global water, sanitation, hygiene, starting with my master's thesis, which was on internally displaced persons and hygiene promotion in the Democratic Republic of Congo. I really had the opportunity to work with amazing colleagues from around the globe in Chad and DRC, and then in global positions in Atlanta and in DC. My experience with WASH has really spanned many different aspects of it, from hygiene promotion, behavior change, capacity strengthening, applied research. More recently, some work with climate adaptive WASH behaviors, WASH in healthcare facilities, and then humanitarian WASH and outbreaks and displacement. One area of WASH that I haven't worked in, I would say, would be in schools, but I've had really the fortune to be on different projects touching many of the different aspects and priorities that we have within the global WASH sector. So when it comes to WASH, we're thinking about water in sufficient quality and quantity, sanitation, which includes infrastructure, services, and promotion, and hygiene, which is both around having an enabling environment, as well as hygiene and behavior change. WASH is so critical because it protects health, prevents disease, and supports dignity. This directly contributes to the Sustainable Development Goal 6 around clean water and sanitation, but also is fundamental to realizing Sustainable Development Goal 3, good health and well-being. And there are specific targets around reducing death and illnesses from hazardous chemicals, air, water, and soil pollution and contamination. And WHO and others specifically track the mortality rate attributed to unsafe water, sanitation, and hygiene under that indicator. In addition to good health, proper water and sanitation services can really support other SDG goals, including no poverty, zero hunger, and gender equality, just to select a few. My first key message is that WASH saves lives. And I think sometimes when we are in the weeds of thinking about water and sanitation, we forget to step back and think about how fundamental this is to good, to health, and how such a basic service can really have widespread health impacts. Water, food, and shelter and immediate health care are the most important human needs in an emergency. Without water, people will die or they will move on in search of it. So you can see here the widespread destruction, for example, in the 2010 Haiti earthquake, and you can imagine how that impacted the water and sanitation infrastructure and access, and how that would then impact health. To understand why WASH is the cornerstone of global health, we can look at this figure from the World Health Organization that centers pressures such as climate change, urbanization, and population growth, which act as drivers for health risks surrounding them. With WASH, we can create a critical barrier against infectious diseases, as shown on the left. We can prevent diarrheal diseases and reduce the reliance on antibiotics that underpin some of the concerns around antimicrobial resistance. Robust WASH infrastructure and monitoring can also help mitigate the chemical risks seen at the bottom. And WASH also supports well-being around dignity, personal safety. Improvements in WASH infrastructure can reduce the risk of injuries and musculoskeletal disorders. There's some evidence that it may support improvements in school attendance. Water is critical to livelihoods and food security. And then finally, it's critical in healthcare facilities, both for infection prevention and control, but also for patient and staff safety, staff morale, and healthcare seeking behavior. People prefer to go to healthcare facilities when they know that there are, clean facilities and there's a place to use the toilets, and they're not putting themselves at risk of exposure to other illnesses. Since 1990, the world has made remarkable progress. The under 5 mortality rate has fallen by 60% and neonatal mortality by 45%. Again, behind these graphs are, an incredible amount of lives that have been saved. And I think in water and sanitation, we could probably do a better job of communicating some of those stories and going maybe beyond these graphs. But I want to highlight these because these gains reflect decades of investment in many global health services, such as immunization, essential health services, newborn care, nutrition support, the integrated management of childhood illnesses, and also improvements in water and sanitation. So just to recognize the work that many colleagues, communities, governments, private sector partners have done around the world to see these incredible results. And it's so important to continue to ensure these basic services are met to continue to reduce the mortality rates. I think it was about last week that the UN released the recent report that estimates in 2024, there was an additional 4.9 million children who died before their 5th birthday. And so despite these decades of progress, we can see that the pace of mortality reduction is slowing, and in some places it's stalling, with the heaviest burdens falling on sub-Saharan Africa and Southeast Asia. In particular, WASH is important to addressing some of these causes of death. You can see diarrhea jumps out in orange, but also WASH is important for sepsis, for malnutrition, and for malaria. We know that also, for example, for acute respiratory infections, simple interventions such as hand washing with soap can reduce respiratory infections by about 17%. And then in healthcare facilities, hand hygiene can prevent up to half of infections. So these basic low-cost services are important in many settings and are also key to disease outbreak prevention and response. My second point is that access to water and sanitation is a basic human right. In 2010, the United Nations General Assembly explicitly recognized the human right to water and sanitation and acknowledged that people need water and sanitation to be able to realize their other human rights. Governments are the main providers and or regulators of water and sanitation in most settings, and these public systems and investments are key. And there's a commitment to this through this General Assembly resolution. Despite the recognition that WASH is a fundamental human right, globally, 1 in 4 people lack safely managed drinking water, 2 in 5 lack safely managed sanitation, and 1 in 5 lack basic hygiene services. And we can think about the importance of improving infrastructure coverage to realize those health benefits, but it's also important to focus on supporting behavior change. Estimates by CDC indicate that on average, healthcare providers clean their hands less than half the times they should. So an effective water and sanitation intervention needs to both consider the infrastructure and behavioral improvements. My third point is that water systems are protected by international law. Damaging or cutting off water supplies is prohibited under international humanitarian law. However, increasingly, water systems are becoming both casualties and weapons of war. For example, if an attack cuts powers to a chlorination plant, treated water cannot be delivered, which then exposes communities to disease. When that water system then fails, hospitals can't maintain hygiene, which then has ripple effects on health. So we cannot discuss the importance of WASH impacts on global health without addressing this weaponization of water. As the Geneva Conventions indicate, infrastructure, services, and civilians providing these services are protected by international law. However, the water conflict chronology shows that water-related violence has drastically increased in the past 10 years. We've seen this recently in conflicts such as Gaza, where UN experts reported last summer that close to 90% of water infrastructure had been damaged or destroyed. And so using thirst as a weapon is against the Geneva Conventions, and this has fundamental impacts on health. My next point is that people really matter. Throughout my career, I've worked with, as I mentioned, really incredible colleagues and community members. At the same time, global shortage of WASH workers is a barrier to achieving the Sustainable Development Goal 6. The 2025 UN Water Report indicated that very few countries have enough skilled personnel to perform key functions. Less than a third reported having sufficient human resources for drinking water, sanitation, and hygiene. In addition to qualified staff, community engagement and planning with people is also very important. We know that building infrastructure is just one aspect, but making sure that WASH solutions are designed with end users in mind really shifts and helps support sustainability. So looking to the future, there's many challenges to achieving the Sustainable Development Goal 6 around clean water and sanitation, including climate change, an increased number of humanitarian emergencies, as well as a deprioritization of public health funding. However, there are many opportunities for innovation in WASH, and we can realize a lot of those health benefits through some of the most basic interventions that can have massive impacts on the lives of kids and people in many different settings. I think I'll stop there and turn it over to Trina.
Thanks so much, Nicole. I love the connection back to what Heather mentioned about the co-development and the importance of involving folks who are impacted in how to solve those problems. So thanks so much. All right, we're gonna go now to Matthew, and looks like your slides are sharing fine, so I'll turn it over to you.
Thanks so much. And I think this is what I'm going to talk about is a nice mix of what Heather and Nicole just spoke about. So I'm going to focus on one aspect of what I do, which is thinking about how we intervene to control enteropathogen transmission in low-income contexts and countries. So I should say my background is I have a PhD in infectious and tropical diseases and public health master's in global and environmental health. And so I'm not a pure epidemiologist. I only play one on TV, but I use a lot of implementation science and field trial work to develop and test interventions and try to understand how we can limit flows of pathogens from sources to people. So back in 2000, there was a review that was somewhat seminal that tried to identify how we address domestic hygiene and diarrhea. How do we reduce some of those diarrhea-related morbidities and mortalities that Nicole was mentioning? And there's a quote here from that review, and Valerie Curtis and Sandy Carencross are some of the giants in the field. And they mentioned human stools should be regarded as public enemy number one. And so in the sector that I work in, the WASH sector, WASH and health, we really think of preventing stool or preventing feces from getting into a child's mouth. And we do that by thinking about what we call the F diagram. And it's not that fancy. It's called the F diagram because a bunch of words that start with the letter F. So we think about fluids, so water, fields, crops, fingers, fomites, which are just kind of any substance, surfaces. And that takes up most of the thinking, I think, in the WASH sector. Talking about WASH systems, water sanitation systems, we're thinking mostly about domestic hygiene, about drinking water, and we can prevent the flows of feces into some of these places by targeting sanitation. And those of you in the United States, maybe you don't realize how magical toilets are, right? In your home and in your school. I mean, it is incredible that you can go into a room in your workplace where you play, where you live, and basically flush your waste out, and you never think about it again. But there's complicated engineering processes that keep the pathogens within those stools sequestered from other people, right, through treatment facilities. So that's a primary barrier to prevent feces from going into the environment. We can wash our hands, and again, in the United States, sometimes we forget how magical it is that we can turn the tap in our kitchens, in our bathrooms, and we can get enough water that's clean that we can use to wash our hands, to wash our surfaces. And we can wash our hands with soap, and hand washing with soap is one of the most cost-effective public health interventions and behaviors out there. We also have clean water, and clean water helps us facilitate that hand washing. And when we drink water from the tap, we can be very, very confident that it is clean, but that's not the case in most of the world. So we have these WASH technologies and services and behaviors that were just mentioned in order to have multiple pathways that break the transmission. And one that is kind of often not focused on in the WASH sector are food and flies. So flies can land on feces that are not contained, and then those flies can land on food. And then all of these pathways can lead to contamination of food. And foodborne pathogens are the largest driver of diarrhea or enteric pathogen morbidity in developed countries. So in the United States, when you think about all these pathways, it's really pathways through food that drive most of outbreaks. And you've heard of many potential outbreaks of leafy greens or peppers or meat. But we also often don't think about the role of animal waste. And so I mentioned before that human stool was priority number one, but actually animal waste is also a major contributor. And it is a major contributor, especially in low-income contexts, low-income countries, where there is not as much of a divide between the domestic environment and animals. Animal foodborne pathogens can also be a problem when you have not just contamination of fields, so the use of animal manure to grow your crops, which is often a concern in low-income contexts because animal waste is a good source of a fertilizer to grow your crops, but also through the slaughtering process. So unsafe slaughtering of animals at home or in markets can directly contaminate the food system. So this pie chart shows the attributable fraction of diarrheal deaths in children by different taxa of pathogens. So the yellow are viruses, the green are protozoa, and the blue are bacteria. And we estimated that around 28% of the pathogens, enteric pathogens that cause morbidity among humans, could be transmitted via animals. Now, we don't really know. There's not really good estimates of total foodborne morbidity, and it's being worked on by the FERG and WHO. They're trying to develop better estimates, but it's still unknown. This map shows the world, and the darker purple shows where there is a larger proportion of animal waste compared to human waste. So in Australia, in South America, in parts of sub-Saharan Africa. And I just want to pause for a second. Everyone think, over globally, what's the comparison of animal waste to human waste? Is it 1-to-1, meaning it's the same amount of human waste to animal waste? Is it half? So there's like half as much animal waste or human waste? I don't have a way to do a poll here, but think about what your guess would be. The answer is 4 times the amount of waste that come from animals per year than humans. So I want to say it's important to think about human waste, but also all this animal waste. If it's untreated, it can be a major concern. So there were 600 million foodborne illness cases in the world, estimated in 2015, and most of it occurred in African countries, so in low resource settings. I'm going to talk about one of our projects, which is the Cheep Cheep study, and it stands for Chicken Exposures and Enteric Pathogens in Children Exposed Through Environmental Pathways. And we are looking specifically at the poultry value chain in Mozambique and in Maputo, Mozambique, with several partners, the INS Instituto Nacional de Saúde in Mozambique, Eduardo Mondlane University, and the University of Washington, along with Emory University. And this has been a multi-phase, multi-year project. And we started with the premise that public markets and the value chain of chickens as they flow from through public markets and through domestic environments should be a major source of concern. And here's some photos of some of the public open air markets in Maputo. On the upper left actually is a smallholder farm where they have raised chicks, and the rest of the photos are from various markets where chickens are brought live, sold live, slaughtered, and then sold for meat in these public markets. And so public markets are really important for livelihoods and for balanced nutrition in many parts of the global South. And so in no way am I saying we should not have public markets, but public markets are largely unregulated and are a major source of potential pathogen exposure for the population. So when you think about a chicken value chain, you have day-old chicks that grow up to then lay eggs, and then you have these broiler chicks that are then sold live or as meat. And so as part of this study, we did mixed methods research to track where these chickens, these eggs, and this meat goes. So from the breeder farms to the hatcheries, to the outgrowers, to commercial abattoirs that then make it to grocery stores and restaurants, or from these small holders that raise maybe 500 chicks. Those might go to the commercial abattoirs, or they might go to public markets where they're sold and slaughtered. So let's follow the chickens. So when you have the day-old chicks here, we found none of them were contaminated with Campylobacter or Salmonella, C or S. So Campylobacter and Salmonella are chicken-related pathogens that are major drivers of morbidity among children. When the chickens went to small-scale farms, we measured that 61% of them were then carriers of Campylobacter and 0% were carriers of Salmonella. And this represents an opportunity for exposure of young children, but also of leafy greens and other farm products. At grocery stores and restaurants, that number of contamination went up to 86% for Campylobacter and 26% for Salmonella. When they went to the live markets, the feces of the chickens, 100% of them were contaminated with Campylobacter and 23% with Salmonella. And the meat was 100% with Campylobacter and 17% with Salmonella. So that's pretty scary stuff. And so our next phase of our intervention is that we've partnered with an architectural firm, and we are doing a study upgrading one of the markets in Maputo using a process of working with the vendors, because if we didn't get the vendors on board with our intervention, there was going to be no intervention. So a lot of work with the vendors to develop and understand what they needed to make their sales work, but also what their process was for slaughtering and selling, and then creating a clean process so that we can have open air public markets that were good for the vendors in terms of their income, but also improved the food hygiene chain. So that's all I have. Handing it back over to you all. Thank you so much.
Thanks, Matt. Great presentations. I love the threads that are running through them. And we do have a few questions, and it's okay, I think, if we go over a little bit. I mean, folks can run away if you have to. We can start with a question for Heather. So Heather, how do you plan to identify which specific species of organisms that were present in animal and humans in Madagascar to see if the source tracking, if they're the same across the lemurs, humans, and livestock?
That's a great question. Thanks, Trina. So we did preliminary screening. We're in very, very remote areas of Madagascar, so no electricity, no running water. And so we did microscopy in the field, which we can identify kind of the organism, but we can't say the species. And then we also tried to extract DNA in the field to then do PCR. So we've identified kind of at the generic level what these organisms might be, but then we're going to sequence select samples to then identify if it's the exact same species that's shared across our different animal species, essentially. Yeah.
Very cool. Thanks. Question for Nicole, and this is a great one. So, given all the different ways that you've worked in this field, what kind of advice would you give students who might be interested in future careers that are similar to yours?
Yeah, thanks for that. I think first, WASH is very interdisciplinary, so there's a lot of different backgrounds that we require. And I think also getting exposure to your problems that might be in your community. You mentioned in your presentation a lot of water quality issues around Wisconsin, so that could be an option. And then also building out your soft skills around communication, facilitation, negotiation, those are also really critical.
Yeah, it comes down to the people, right? Matt, what is causing the increase in the Campylobacter as the chickens move through that food chain flow?
That's a great question. So I think when they get to the smallholder farms, it's the lack of hygiene between the flocks. So you have 500 chickens in an area, you sell them all, and then the lack of cleaning means that there's residual chicken feces in that area. Then when they move to the markets, they're basically stacked on top of each other in close quarters, right? And so as we know, from COVID, this is not just a problem for enterics, this is a problem for potential respiratory viruses. So yeah, it's about the lack of hygiene within the chicken storage areas. So for example, one of the things we're doing in the markets, renovation, which is almost done. It's happening right now. We're creating drain systems so that and water systems so that they can be easily washed. The surfaces can be washed, and in between the kind of the new batches of chickens, the solid waste can be removed. And then the last step would be from the live chicken to the meat, and that has to do with the slaughtering. So when chickens are slaughtered and eviscerated, they are contaminating themselves, and there's a lack of water in these open air public markets. And the chicken, we actually did a study looking at the water. The water is clean when it starts off, and then the moment you put in the first chicken after eviscerating to wash, that water is fully contaminated, and the people that do the slaughtering don't change out the water. So you're basically contaminating the meat each time. And so if you can imagine every chicken that leaves the market as meat is fully contaminated with Campylobacter, and that chicken is going home. And of course, if you cook it, you will kill the Campylobacter. But in the meantime, all the fomites, all the touching in the house, that's potential source of contamination. And just to add, in our study, 86% of children 1 year old had at least one pathogen at 1 year. So that's an incredibly scary amount of pathogens in 1-year-old children.
Wow. Yeah. So it all comes down to the water, right? Okay. Well, so one last question was for me, and it was about what kinds of lakes are most impacted by the blue-green algae blooms. And I guess I didn't want to imply that it was just a problem in the US. We have blue-green algae blooms all over the world, anywhere humans are coming into contact with freshwater bodies. So a lot of issues with cyanobacterial blooms would co-occur with some of the pathogens that my fellow panelists have talked about as well. So all these different potential threats that we have to think about in a systems level way. So I think we are a little past time. So I am going to hand it back to Kaylin to wrap it up. Thanks.
Great. Thank you all so very much. This was absolutely wonderful. And unfortunately, that is all the time we have for today. So on behalf of the University of Wisconsin-Madison and our entire global health community, thank you to Trina for moderating this engaging discussion, and to you, Heather, Nicole, and Matthew, for sharing your impactful work and insights. We are very grateful for your leadership and appreciate your participation today. And we will be pausing our webinar series in April as we are co-hosting our annual Global Health Symposium on April 8th, titled Establishing Trust for Emergency Preparedness, the Role of Universities and Alliances. So please register. You can join us in person or online. Our last webinar series of the academic year will resume on Tuesday, May 19th, with a session on cannabis and One Health, connecting people, planet, and policy. Please visit our website at ghi.wisc.edu to learn more about these upcoming events and to access today's archive video. Thank you, everyone, and have a wonderful rest of your day. Thank you.